तो अब हमने जो प्रोसेस अडॉप्ट करना है उसके लिए वी ऑलरेडी टॉक्ड अबाउट द reagents and supplies that is going to be used for ultrafiltration of amplified products. इसके लिए हमें एक कंसंट्रेटर जिसको हमने सेंट्रिकॉन U100 का नाम भी दिया, concentrator, then some tubes are required. The process is very simple, that we are going to load 2 milliliter of TE buffer. जैसा कि पहले भी बताया, TE buffer is meant to dissolve the DNA. The 2 ml is going to be applied on the reservoir chamber, and then we have to take our amplified PCR product. अब यहाँ पे इस चीज़ का ख्याल रखना है कि अगर हमने एम्पलीफाइड पीसीआर प्रोडक्ट को जब सेटअप किया था, पीसीआर रिएक्शन को, अगर उसमें हमने ऑयल यूज़ किया है और नहीं किया, इफ़ वी हैव यूज द मिनरल ऑयल, then we have to remove the mineral oil and we have to take only the amplified PCR product from the previous PCR. If you look at this picture, this is the concentrator. This concentrator is going to be filled up with the 2 milliliter TE buffer और फिर इसमें हमने लोड करना है अपना PCR amplified product. This is going to be placed in a collector tube, and this unit, the complete unit, is going to be placed in a centrifuge. The entire unit is placed in a centrifuge for centrifugation. इससे होगा क्या कि आपका जो TE बफर और जो लोडेड सैंपल है, that will pass through the ultra filter. जो unwanted component हैं, that contains the primer, that contains the DNTPs, they will be passing through the filter, और जो amplified DNA है, it will remain associated with the filter. We have to centrifuge this loaded concentrator for 30 minutes at 100 G speed at 4 degree centigrade or in between 4 to 25 degree centigrade. इस 30 minutes period में all the liquid and unwanted components, they are going to be filtered, and whatever left behind with the filter paper or the ultra filter paper, that is going to be recovered subsequently. Once we have done with this centrifugation, the concentrator is separated from the collector's tube. अगली स्लाइड में एक पिक्चर में देखेंगे, और उसके बाद इस कंसंट्रेटर को हम प्लेस करेंगे एक नई ट्यूब में to collect the purified product. For that purpose, we are going to invert the concentrator and we are going to place it in the collector or retentive cup, and again we are going to subject it for centrifugation for 2 minutes. अब इस स्लाइड में अगर आप देखें, कंप्लीट प्रोसेस को 4 डिफरेंट स्टेप्स में शो कर दिया गया है. दिस इज द ट्यूब फॉर कलेक्शन ऑफ द वेस्ट. दिस कंप्लीट यूनिट is the concentrator tube or concentrator cup. When we add our sample here, we spin it or centrifuge it, all the waste is collected here. The DNA purified sample remains in the concentrator cup associated with the filter. In the next step for recovery, we have to use this concentrator by in inverted position like this one. In this figure you can see that we have inverted this concentrator and we simply subject it for centrifugation for 2 minutes. This centrifugation has collected the DNA which is purified and which was associated with the concentrator is now separated. Now this concentrator is no more required, it can be discarded. Our required purified DNA sample in this collector's tube. If we want, we can precipitate this sample by adding sodium acetate and ethanol if it is required. This is not mandatory because this is already purified by the concentrator, and If the concentration is not yet reached to our required level, we can this process again and again until the concentration of purified DNA is reached to our required concentration.